When you think about how life has evolved on Earth, we need like a helpful structure to kind of put this evolutionary history into. If you've gone to a few biology classes, you've probably seen a tree structure. That tree structure is usually simplified when you see it, but it's very helpful as like a general descriptive for organizing the relationships of one organism or species to another. While being very helpful for research, it's not always correct.
One of the primary goals of JGI is to sequence novel organisms and determine taxonomically how they fit on the tree of life. And the more organisms get sequenced and the more complete the tree of life is, the easier it is to place new organisms on it, usually, unless there are mistakes. If there are mistakes, then everything you build on top of those mistakes is also going to be wrong.
And so my project is concerned with writing software that's going to go in and make us aware of these problems, and so we can hopefully fix those. The taxonomy that we're talking about for my JGI project definitely includes all of life as we know it. That's, of course, an incredibly vast amount of information. It's one of those numbers that's kind of difficult to think about. We have a taxonomy and then the similarity numbers for every species in that taxonomy. Similarity numbers are just a sequence to sequence, genome to genome comparison. So if you have two identical genomes, then they're going to be identical. Their similarity is 100%. Whereas like two really disparate genomes are going to be, 50% lower, maybe not even comparable. They may not even have sequences in the same configuration. And so what we want to do is go through the taxonomy and compare the tree relationships to the similarity scores. And so we can figure out like, hey, there was a misplacement for this particular organism in the taxonomy. We need to move that somewhere else in the tree. The importance of this is that other biologists are using this taxonomy as a reference for their own work. So if you want to, say, find the closest related organism to a disease so you can experiment on something that's maybe not as pathogenic, you have a better idea of what the closest related organism is. And so learning about how to manipulate these trees and these structures it's super cool. It pushes into like what I'd like to continue working on with the rest of my career, and it's super useful for all of biology.